Microfluidic technology offers a highly efficient and cost-effective way for molecular diagnostics compared to traditional methods. This is especially important during disease outbreaks such as the COVID-19 pandemic caused by severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2. Microfluidics utilizes portable devices ranging from small lateral flow strips to benchtop analyzers that conduct tests near the patient, reducing the need for transportation of samples to a laboratory. One of the primary benefits of microfluidic technology is that it simplifies or eliminates the need for complex sample preparation, rapid signal amplification, and sensitive signal readout. As a result, test results can be generated accurately and quickly within minutes instead of days. The widespread availability and production of microfluidic-based point-of-care tools have expanded their applications outside of hospitals and clinics, even allowing for tests to be performed in a patient's home. The core concept behind microfluidics is a miniaturized analytical platform called a lab-on-chip or LOC. The LOC integrates several laboratory functions including sample preparation, processing, and analysis onto a tiny chip or substrate. The LOC's goal is to reduce costs, analyze time and reagent consumption, while also improving the accuracy, sensitivity, and selectivity of analytical assays. Microfluidic devices were first introduced in the 1980s, but it wasn't until soft lithographic techniques were developed in the 1990s that the study of lab-on-chip or LOC began to progress rapidly. Soft lithography is a method of creating tiny channels and structures on the surface of a chip using pliant materials like polydimethylsiloxane or PDMS. This technique has made the production of intricate microfluidic devices affordable and scalable by allowing precise control over channel geometry and dimensions. Since the introduction of soft lithography in the 1990s, the LOC field has expanded significantly, with more than 2,000 papers published over the last 20 years. Lab-on-a-chip, or LOC, has diverse applications, including nucleic acid biotechnology and analysis, such as DNA and RNA sequencing, genotyping, and gene manipulation. It also has applications in protein analysis, including proteomics and metabolomics for both targeted and global analysis. Medical diagnostics, including point-of-care and molecular diagnostics, as well as medical devices and treatments, including implantable and wireless devices, are other areas where LOC is used. Additionally, LOC plays a role in drug development for screening and delivery, cells and tissue engineering, single-cell analysis, systems in synthetic biology and medicine, and energy and environmental monitoring for health and security. Due to these advantages, microfluidics have been one of the top 10 revolutionary technological candidates for some time now. In the field of nucleic acid analysis, LOC is a popular and versatile tool. LOC devices work by using well-designed geometries and various physical effects, such as pressure gradients, capillarity, electrokinetics, magnetic fields, and sound waves to manipulate fluids. However, LOC devices are incredibly delicate, making them difficult to fabricate, package, and interface. Additionally, achieving multiplexing and reuse can also pose a significant challenge. Despite these limitations, LOC offers several unique advantages over other platforms, including maximum application diversity and excellent compatibility for technologies. Within microfluidic chambers, fluids flow in a highly organized way. Specifically, they can flow in a layered or laminar manner, with the streams remaining parallel to one another. This means that there's no mixing between the streams. Alternatively, when fluids move in a near-random manner due to various inertial forces, it's called a turbulent flow. Technicians try to avoid turbulent flows because they can affect reading accuracy. When we talk about liquids flowing in very small spaces, like ones that are about 1 micrometer in width or length, the flow tends to be laminar. This means that when two streams of fluid come together in a microchannel, they end up flowing side by side, making the mixing of fluids a little more complicated. But the predictable and controlled nature of these fluid flows provide microfluidics with special mechanical advantages. Moreover, microfluidics offer economical benefits by allowing us to shrink and integrate multiple components at scale. The microfluidic chip has different functional components like valves and pumps that work together to manipulate the nucleic acids during amplification. The chip can be designed to automatically perform multiple actions, reducing the time and effort needed for manual sample processing. Let us go through the process of using microfluidic chips to analyze nucleic acids. First, the nucleic acids are taken from the sample and purified in different ways, depending on the type of sample and downstream application. Then they are put onto the microfluidic chip where they are amplified using polymerase chain reaction or PCR or amplification-free techniques. Before running the test and amplifying the sample, we might need to break down a cell in a procedure called lysing. Then, the prepared sample fluids are moved around the chip through tiny channels, and we might perform laboratory actions like mixing, separating, or filtering. For example, we may mix the prepared sample with various reagents. After amplification, scientists can detect nucleic acids using different techniques. Two commonly used methods are fluorescent-based detection and electrochemical detection. In fluorescence-based detection, fluorescent probes are used to specifically bind to the amplified nucleic acid sequences. These probes emit light when they're excited with a laser or LED. In electrochemical detection, electrochemical sensors are used to detect changes in the electrical properties of the solution when nucleic acids bind to them. After the process is complete, the result is interpreted and delivered to the end user. Microfluidics can have shorter result times because they involve manipulating small volumes of fluids in microchannels, which allows for faster reactions and analysis. The smaller size of microfluidic devices also enhances mass transport and fluid mixing, leading to faster and more efficient reactions. Additionally, microfluidic devices can integrate multiple assay steps into a single platform, reducing the overall time required for sample preparation, processing, and analysis. Microfluidics is more sensitive in testing because it allows for the detection of very small sample volumes with high precision. The small dimensions of microfluidic channels and chambers provides a large surface area to volume ratio, which enhances the interactions of analytes with sensing elements and increases the detection sensitivity. Furthermore, the miniaturization of analytical systems in microfluidics reduces the instrument noise, improves the signal-to-noise ratio, and enhances the overall sensitivity of the system. Additionally, the ability to integrate multiple assay steps in a single device further enhances the detection sensitivity and reduces errors associated with sample transport and handling. The technology offers a quick way to detect genetic markers for diseases or pathogens, diagnose diseases at the point of care, identify cancer, and more. It also reduces the time and resources needed for traditional laboratory analysis, which allows for faster and more efficient high-volume testing of nucleic acids. One of the main advantages of lab-on-chip technology is the ability to analyze smaller sample volumes, reducing the amount of required materials and lowering costs. It is also suitable for point-of-care testing, making it easier and more convenient for patients to receive diagnostic information. This technology is ideal for developing wearables and home testing platforms that can provide individuals with more information about their health. However, several challenges, such as the need for consistent quality in mass-produced chips, limitations of single-use cartridges, and regulatory issues must be addressed to expand the use of lab-on-a-chip technology. Microfluidic point-of-care testing or POCT devices are gaining increased interest from manufacturers worldwide due to their ability to automate tests and require limited reagents. These devices offer a promising future for molecular diagnostics in urgent or daily situations and warrant further research. Since the outbreak of COVID-19, microfluidic devices and newly designed chips targeting the virus have been launched, such as the film array BioFire, Genplex, and Vivalytic. Additionally, sexually transmitted diseases are gaining attention and associated equipment is being created, such as the IO single module system and Visby Medical. Some novel chips are even integrating various techniques, such as smartphones as portable analytical devices and surface enhanced Raman spectroscopy coupled with microfluidic devices. Commercial microfluidic POCT devices strive to match the test sensitivity and specificity of modern medical tests conducted in hospitals and laboratories. In epidemics, these devices are crucial for efficient diagnosis without the need for a large-scale laboratory or testing center. However, there is still room for development in terms of efficiency, automation, integration, sensitivity, specificity, portability, and affordability, indicating that the microfluidic POCT industry is still in its early stages of application. Thank you for watching.